The topic I would discuss here is factors affecting somatic embryogenesis, applications of somatic embryogenesis, and synthetic seed production. In the earlier class, I had discussed about what is somatic embryogenesis and the various stages of somatic embryogenesis. How is it different from zygotic embryogenesis? Here, we would go through the various factors that affect the process or induce somatic embryogenesis. Steps involved in somatic embryogenesis. The explant, after accession from the donor plant, is surface sterilized and inoculated in nutrient media. The explant could be a leaf lamina, petiole, internode, inflorescence to name some. Somatic embryos can be induced by direct or indirect methods based on the media composition. High concentrations of auxins, especially 2,4-D, has been found to induce somatic embryogenesis. Suspension cultures can also be used for somatic embryo production. There are two alternative mechanisms by which an explant can regenerate into entire plantlets: organogenesis and somatic embryogenesis. In organogenesis, shoot, root, or both are formed from the dedifferentiated cells based upon the nutrient media composition. These are formed from unipolar cells which have a vascular connection with the mother tissue and the regenerating tissues. In the case of somatic embryogenesis, somatic embryos occur from single bipolar cells with no vascular connection with the surrounding cells. Somatic embryogenesis is induced in non-embryonic cells by exposure to certain stress, growth hormones, differential intensity of light, pH and heat shock, or certain chemicals. Induction of somatic embryogenesis depends on the genotype, the physiological state, the developmental stage, and environmental factors of the explant. The genotype of the explant affects the presence of endogenous growth hormones. Explants with high level of endogenous auxins are more responsive to somatic embryogenesis. Induction of somatic embryogenesis leads to activation of an array of genes and transcriptional factors which is responsible for embryogenic capability of the cell. It makes the somatic cell competent and then embryonic, leading to the generation of plantlets. The factors affecting somatic embryogenesis can broadly be classified into structural factors, physiological factors, and gene expression. Structural factors. Somatic embryos are formed from single bipolar cells. The initiation of polarity is believed to be the first step towards embryogenesis. In some plants, like carrot, the first division is asymmetric, giving rise to a small densely cytoplasmic apical cell and a large vacuolated basal cell. The smaller cell forms the embryonic cell. In some, the asymmetrical cells produce a mass of pro-embryogenic cells rather than forming the embryo. One of the few pro-embryonic cells would form the embryos and the remaining are removed by programmed cell death. Structural factors like size of the cell, microtubule arrangement, and cell wall influence induction of somatic embryogenesis. Physical isolation of the cell also plays an important role in somatic embryogenesis. Hence, suspension culture is successful in producing somatic embryogenesis in many plant species. Distribution of cell wall surface antigen with epitope for antibody Gim8 has been reported in many cases. After the asymmetric cell is formed, one of the daughter with the cell wall antigen is recognized by the antibody, and the other without it, or the epitope-free cells lead to the formation of somatic embryos. The figure here is a schematic representation of the different cell states which lead to induction of somatic embryogenesis. Cell state A is the normal cell present in explant or callus. It does not have the epitope for Gim8 antibody. When given an external stimuli like high auxin concentration, it may switch to state B, which contains the surface antigen for the antibody. From state B, majority of the cells switch to state D and finally leading to apoptosis and cell death. These two have the cell wall antigen for antibody Gim8. Some cells from state B can move to cell state C, which could be or may be after a asymmetric division. The cells in state C do not have the surface antigen and become the proembryonic cells while still attached to the sister state D cell. Physiological factors that affect somatic embryos are: first, plant growth hormone. Plant growth regulators play a key role in somatic embryogenesis. The presence of endogenous hormones like indole acetic acid favor somatic embryogenesis. High concentrations of auxins in the nutrient media, especially 2,4-D, has found to induce somatic embryogenesis. However, further growth and maturation of somatic embryos occurs in reducing the levels of auxins. Abscisic acid is found to help in maturation of somatic embryos. Gibberellic acid is believed to inhibit somatic embryogenesis but required for germination of mature somatic embryos. Second, carbon source. High sucrose concentration is believed to induce somatic embryogenesis. In citrus species, using glycerol instead of sucrose favored somatic embryogenesis. Concentration of ammonium salts also affect somatic embryogenesis. Casein hydroxylates, proline, and serine have been found to be effective in inducing somatic embryogenesis. Light quality, photoperiod, pH changes or shocks, temperature shocks also affect the process of somatic embryogenesis. Certain additives to the media like antibiotics, ethylene action inhibitors, oxygen concentration also affect somatic embryogenesis process. The cell culture density also plays an important role in inducing somatic embryos. High cell density favors embryonic cell initiation from single cells, but for development and maturation, less cell density is required. This could be due to the secretion of proteins or other cellular factors into the media. Addition of arabinogalactan proteins is found to promote somatic embryogenesis. A temporary desiccation period before generation facilitates regeneration of embryos to plantlets. Many genes are involved in initiation and development of somatic embryos. A total of 51 proteins have been identified that may function in early somatic embryogenesis. These proteins are involved in hormone signal transduction, chromatic remodeling, cell cycle re-regulation, metabolic activities, GTPase signal transduction, transcriptional regulation, meristem formation and maintenance, and apoptosis and microtubule organization. The genes can be classified in three categories based on its function. First, embryonic induction, then second, embryonic, and third, maturation. Many genes have been identified which are responsible for inducing signals for somatic embryogenesis in response to an external stimuli. Increase in signaling molecules like somatic embryogenesis receptor kinase has been reported during somatic embryogenesis. Epigenetic factors like DNA methylation, histone modification, and microRNAs of the explant affect somatic embryogenesis. Hypomethylation promotes somatic embryogenesis induction. The presence of auxins and other plant growth regulators and certain stress conditions induces the expression of several genes like ARF7, ARF19, PRC1, which in turn induces signaling by means of a jasmonic acid pathway. It influences cell metabolic pathway leading to de-differentiation of the cell. After dedifferentiation, the endogenous level of auxin increases, which regulates the expression of many genes like Wuchel genes, CLF, somatic embryogenesis receptor kinases. All this leads to chromatin remodeling, salicylic acid and jasmoic acid signaling pathways, leading to totopatency of the cell. This, in presence of auxin, further activates an array of signaling molecules and gene activation, leading to induction of embryonic cells. In some instances, the primary somatic embryos fail to go for maturation and enter into successive cycles of continuous production of supernumerary embryos from the somatic embryos itself. This is known as secondary embryogenesis or recurrent, repetitive, or also known as accessory embryogenesis. Applications of somatic embryogenesis. Somatic embryogenesis helps in micropropagation of large number of embryos with synchronous development in a short period of time. It is preferred over meristem culture because simultaneous development of shoot and root involves fewer cultures, leading to cost efficiency and reducing time. Bioreactors are used for large-scale production of somatic embryos. Somatic embryos are used for the production of synthetic seeds. This helps in long-time storage of embryos and can be cryopreserved for germplasm storage. Somatic embryogenesis helps in propagating plants which do not have seed set or vegetative propagation. The plants obtained from somatic embryogenesis are disease-free. The embryoic cells can be used as a source of protoplast isolation and can be used to create somatic hybrids or recombinant cells by gene transfer. Certain plant species show recurrent embryogenesis. These offer a great potential for production of seed storage proteins, lipids, and other embryo metabolites. Somatic embryogenesis has been successful in generating somoclonal variants in many plant species. The process of somatic embryogenesis is utilized in basic research as a model system to study the molecular, physiological, and developmental events during embryogenesis. What are synthetic seeds? Synthetic seeds are encapsulated somatic embryos which function or mimic normal seeds and can develop into seedlings under suitable conditions. For production of synthetic seeds, there should be a mass production of somatic embryos, and then these are encapsulated to produce synthetic seeds. Based on the encapsulation done on somatic embryos, the synthetic seeds are divided into two types: desiccated synthetic seeds and hydrated synthetic seeds. Desiccated synthetic seeds are somatic embryos which are encapsulated in polyoxyethylene glycol. These synthetic seeds are produced in desiccation-tolerant plant species. Hydrated synthetic seeds are seeds in which the somatic embryos are encapsulated in hydrogels like sodium alginate, potassium alginate, or combinations with gelatin, agar, in certain cases, gorgum. In this process, the somatic embryos are first coated in alginate solution and then dropped into concentrated calcium chloride solution for 20 to 30 minutes with slow stirring. The gelled embryos are then washed with water and then can be stored or inoculated. The principle behind encapsulation of somatic embryos with alginate matrix. Here, the sodium alginate droplets containing the somatic embryos, when dropped into calcium chloride solution, form round and firm beads due to ion exchange between the sodium ion in sodium alginate with the calcium ion in calcium chloride solution. For large-scale production of synthetic seed, automated encapsulation processes are also available. The figure here shows the production of somatic embryos followed by excision of somatic embryos and encapsulation of these somatic embryos by alginate and sodium chloride method. The synthetic seeds then formed are allowed to germinate to give rise to the plantlet. Thank you.